We could use a wide variety of techniques for monitoring protein denaturation depending on the denaturation factor such as denaturants, pH, and temperature. Due to the numerous amounts of techniques, we will only enumerate one of the most recent methods used in literature. You may notice that the overall principle remained the same among all these methods, wherein you monitor a certain characteristic in the protein that is different in the native and denatured state. So in Park et al., they use CD spectroscopy to monitor change in secondary structure, and dynamic light scattering to measure the changes in hydrodynamic diameter. For Du et al., UV-Vis was used to measure hydrophobicity via the differences in blank ensample and sulfhydryl content via Elman's reagent. Fluorescent spectroscopy was used to monitor tertiary structure via the shift in lambda max and intensity due to the exposure of tryptophan residues. FTIR was used to monitor the secondary structure by looking at the distinct peaks at the alpha helices and beta sheets. For Bigar et al., qT-PCR was apparently used. They monitored the change in the binding to the fluorescent dye. As more hydrophobic residues interact upon denaturation, more binding towards the dye takes place. And lastly, for Jelinska et al., Taylor dispersion analysis was done. Mainly, it was used to monitor the diffusion behavior.
Basically, in Anvindsen's experiment, 100% recovery of the enzyme's function was not obtained, primarily due to randomly formed disulfide bonds even in the absence of urea. Urea formed unwanted hydrophobic interactions at the core of the enzyme. This essentially affects the folding of the protein, where the hydrophobic residues buried deep at the core will sort of attempt to have hydrophobic interactions with urea. This will eventually result in the scrambled structure with the corresponding loss of function. When the denaturant urea is removed and mercaptoethanol is still present at trace amounts to reduce the disulfide bonds, these disulfide bonds can still even randomly form. For instance, we do not know if during refolding, the disulfide bonds are actually correctly formed similar to the native structure, because the disulfide bonds may have a preference for binding with the thiol group found at the surface residues instead of those found in the hydrophobic core. Especially during Anfinsen's time, they may not have any access to techniques that can determine if the disulfide bonds correctly bonded. Eventually, this can result to the protein misfolding, where the configuration of the protein structure would be different with the native state. Maybe, for instance, if X-ray crystallography is available, they can perform this method to check if the structure is similar or not by comparing, let's say, if the matrix or configuration spaces are similar, to arrive at correct disulfide connectivity. So here's the question of Shane. You mentioned that a factor for optimal protein renaturation includes amino acids not forming new bonds. May I ask you to expound on this? Say, what kind of new bonds can amino acids make during the time it is denatured and then renatured? So for that, we want to avoid the formation of new bonds such as the formation of disulfide bonds, as this would affect the structure and consequently the function of enzyme. It can even lead to the loss of its function. Basically, we can think of these bonds as sort of sticky glues that when given the opportunity to interact, they can form unwanted new bonds. We can also think of other interactions such as ionic interactions and hydrogen bonding if, say, the pH of the solution is not carefully assessed and can result into protonation or deprotonation of some residues. This can consequently lead to the said sticky bonds, and when not resolved, can result in the formation of a random structure. I hope this helps. Let me know if. For Marian's concern, yes, they will always denature at higher temperatures, especially at really high temperatures. Basically, we learned that there are levels of structure in proteins. Denaturation can result into the disorganization of the protein's native structure, particularly the quaternary, tertiary, and secondary structures. However, this excludes the primary structure, which is essentially the amino acid sequence itself. On the other hand, do you know that you can actually denature a protein at very low temperatures as well? This is due to the general phenomenon caused by the very specific, strongly temperature-dependent interaction of protein nonpolar groups with water. For Louis' question, we learned from Chem 33 or organic chemistry that the structure of molecules affect their physical properties, such as their boiling point, melting point, and freezing point. By this logic, we can think of for amylopectin, the branch chain of monomeric glucose, as the one, for instance, having a lower boiling point, as they cannot pack together well, primarily due to its branch structure. We can credit this to IMFA, such as van der Waals forces of attraction and hydrogen bonding. This can also give rise to the observed formation of a gel when you increase the temperature of a solution containing amylopectin. On the contrary, this is not observed for a solution containing amylose. But to discuss only the physical properties would be limiting. The structural differences actually have effects on their metabolism and release of energy. For instance, amylase can act effectively on amylose and hydrolyze the straight chain with the accompanying release of a glucose monomer. On the other hand, amylopectin would require certain debranching enzymes to effectively hydrolyze the branch linkages. In this way, other hydrolase enzymes that can now act upon the alpha-1,4-glycosidic band of the polymer can now perform the hydrolysis and the eventual release of glucose. I hope this helps.
Why did we use 0.8 NaCl solution for the dilution of human saliva? So NaCl is used in order to activate the salivary amylase. But what exactly activates the salivary amylase? It is due to the introduction of Cl ions. So why 0.8? The 0.8% is the concentration that induces the highest enzymatic activity. It also replicates the physiological conditions. And next is the sulfuric acid. Why did we use sulfuric acid for the termination reaction? Sulfuric acid induces the protonation of residues, leading to the alteration of charge, thereby leading to the alteration in the interactions of residues. We can see this alteration in the effect of pH. However, the addition of sulfuric acid is at a larger extent since the solution is concentrated, leading to the immediate termination of the activity. So we have mentioned in the video that we use the words optimal temperature and optimal pH, but are those the right terms? Although it is true that the determination of optimal temperature and pH is not the goal of the portion of the experiment, it provided a point of comparison in the changes in behavior between the denaturation and renaturation. With showing the changes in values of the lowest absorbances, we can observe that not all denatured proteins are renatured back. Another term might be better used is the highest enzymatic activity or lowest absorbances, as it is more general and does not incite comparison on literature values.
From the results, initial denaturation of alpha amylase results in decreasing trend of activity as observed with increasing absorbance. However, upon denaturation with an acid, the enzyme gradually recovers its hydrolysis activity as observed from the decreasing absorbance. Ideally, the absorbance would be similar or close to the initial value. However, from the results, a higher absorbance was observed, indicating of a lower hydrolysis activity of alpha amylase. Of course, we can incorporate some errors in this observation, but the result would also indicate or suggests that a partially unfolded intermediate state must have been formed from acid denaturation, where the immediate state has a lower activity compared to the initial state. The intermediate state may have been a result of residual structures or incomplete denaturation of the structure of the enzyme. We can infer from this that there are some residues of the enzyme that were protonated. which results in conformational changes in the structure. This suggested that there are certain domains in the enzyme that are relatively less stable to facilitate the formation of another state. In fact, this observation is actually similar to the findings of Singh and colleagues, where they found out that a partially unfolded intermediate state was formed upon acid denaturation of alpha amylase. These findings contradict the literature reports that amylase, particularly in the amylase enzyme found in soybeans, follow a two-state folding mechanism. Their study suggested, however, a non-coincidental transitions for alpha amylase, which indicates the presence of intermediates as a result of perturbations of domains at specific intervals rather than simultaneous transitions.